Nutrition and physical activity research is challenging to do well and can be quite challenging to interpret. Now let's look at why. Bias and conflicts of interest aside, we've covered those. Nutrition research is not easy to do well precisely because human physiology is complex, and what we eat is only a small part of the story. What our body does with what we eat is just as important, if not more important. What you see on the top left is a mitochondria, often referred to as the powerhouses of our cells. Power generators is probably the fairest description, as these very specialized organelles are needed for aerobic energy generation. In other words, they break down fats, glucose, and ketones to produce ATP, the energy currency of our cells. But we aren't all blessed with the same abundance of these mitochondria. Think back, and you'll hopefully remember that one thing physical activity does is stimulate the production of mitochondria, because more mitochondria means more energy generated faster when we need it. Another way people differ is their genes, their DNA. Some of us are more prone to conditions that result in poor bowel health and the reduced micronutrient absorption that often follows. Others are born with better protection against oxidation and therefore can get away with less of the antioxidant vitamins in their diet. Others are poor at converting provitamin A, so for example beta carotene, into actual vitamin A, which we can only get from animal sources. And therefore, people who are genetically prone to making the conversion poorly would struggle on a vegan diet, whereas others wouldn't have that particular problem. And the list goes on. Our gut microbiomes differ. This means that even genetically identical twins can respond quite differently to the same foods. The bacteria living inside our bowels do everything from help us break down food by producing enzymes we can't, to producing vitamins that we need, protecting us from infection, and producing neurotransmitters which can actually communicate with our own nervous system. We are very closely tied with these mostly bacteria, but also fungi and viruses that basically call our insides home. And let's not forget, our thoughts change our physiology. Two branches of our nervous system largely oppose each other, one being the sympathetic nervous system, often referred to as the fight, flight, and free system, and the other being the parasympathetic nervous system, otherwise known as the rest and digest system. It should make sense, given the name, that when we eat or have just eaten, and that's actually quite a long period during which we digest, many hours, we ideally want to be in the state where the rest and digest system is dominant. Recent physical activity, as discussed in other modules, also has a big effect on physiology and heavily influences what our body does with the nutrients that we eat. Then there is also the timing of nutrients. For example, our bodies are better at handling carbohydrates at certain times, but this is somewhat individual and we have more to learn about it, so I won't say categorically a certain time is better for every single person, but we know there is a rhythm to these things, and each of our rhythms is slightly different, basically along the lines of us being more a morning person or more a night person or somewhere in between. We'll also look at another aspect of timing in a future module when we look at time-restricted feeding and also intermittent fasting. And sleep is another major influence on physiology, including how we handle the foods we eat and even what foods we might crave. It's relatively well known that, at least for the majority of people, a poor night's sleep results in craving more calorie-dense foods, more fatty foods, more sweet foods. And these are not all just dials that affect our metabolism pulling in different directions. They all affect each other as well. Physical activity influences the mitochondria, for example. It influences the gut microbiome. It influences our sleep, and it even influences which genes are more or less expressed. Sleep in turn affects how well we cope with more intense or prolonged physical activity, as well as changing gene expression and so on. It's a very intricate dance. Yet most studies will measure very few of these. Think back to your own data collection. You had at best a reasonable estimate of your food intake and a crude guesstimate of your physical activity. A lot of nutrition studies don't even have the physical activity estimate, and vice versa. Physical activity studies may not even have the estimate of nutrient intake, or if they do, they might only really focus on total calories or macronutrients. I and a few colleagues are trying to change that, but for now, most studies simply cannot measure the influence of so many factors. Let's stay with the effect of nutrients on genes, or rather genes on nutrients in this case, for just a moment. If you have an interest in the area, you might have noticed that one study touting the health benefits of coffee is often followed by another saying almost the opposite. I used to think this kind of contradiction was basically down to sheer incompetence. It turns out I was only partly right, because not all of us are equally good at dealing with coffee. Some people deal well with coffee and are therefore likely to reap some benefits from having it regularly. Others deal poorly with coffee and would be better off not having it at all. If two studies had very different proportions of each of these types of people, they would come up with different results. But that's because they asked the wrong question. They asked, is coffee good? Well, it may not have been as simplistic as that, but basically that's what they were getting at. Maybe they asked, is coffee good for? What they should have asked, and now there's really no excuse that there are still studies coming out looking at this in a crude way and not factoring people's genes into it, what they really should be asking is, who is coffee good or bad for? So with research, not just nutrition, physical activity research, with all research, the quality of the question basically determines the quality and usefulness of the answer. And researchers, sadly, are still often asking pretty poor questions. And nutrition and physical activity research are far from immune here. And if you think back to the module on conflicts of interest and biases, you can probably see at least some of the reasons why. Beyond that, think back to making your own one-day nutrient reference value compliant meal plan. You had to consider 6 macronutrients. If we call energy a macronutrient, it's not obviously a nutrient as such, it's something we derive from some of our macronutrients. And the number of macronutrients would go up to 7 if you had alcohol, because it also provides energy. Now add to that several vitamins, which you looked at, minerals and trace elements, although not all of them were included. And consider that, as you learnt in the modules related to vitamins, minerals, mostly vitamins though, many of them had different forms that in turn have different bioavailabilities and even different functions in the body. And you didn't even consider the numerous non-essential nutrients, nor did you consider the many toxins we're exposed to in our diets, like heavy metals, herbicides, pesticides, antibiotics, etc., depending on what and where you eat. And although you did collect some physical activity data, all the nuances of that were essentially removed by converting everything into minutes. And there was no objective measure of physical activity either. Again, that's why I called it a guesstimate. The vast majority of nutrition studies focus only on the macronutrients. To be fair, when a diet is prescribed as part of a study, dietitians are often employed to ensure the diet meets micronutrient guidelines, like the nutrient reference values. But that's almost more in theory. They actually do that. But what participants do in practice, interpreting the instructions, is a whole nother matter. Now, of course, we have any number of wonderful applications, and I do think Chronometer is certainly among the best, if not the best. So maybe studies will start changing a little bit in this regard. Let's get back to more the basics for a minute. We simply cannot achieve the scientific ideal of changing only one variable when it comes to nutrition or even physical activity. So we therefore cannot easily say that a given outcome was because of a specific change. Let me show you what I mean. If we add more protein into the diet, or more carbohydrate into the diet, or more fat into a diet, well, total energy goes up. So we've still changed something. We've changed energy. What would you expect to happen if we give any one of these in greater amounts to someone? They're probably going to gain a degree of weight, right? We would expect that because we've changed energy. Also, the ratios change. If in a given diet I add more protein, well, suddenly the proportion of protein will be higher, and relatively speaking, the proportion of carbohydrate and fat will be lower. There's always going to be a change. If we keep the energy the same, then the ratios change even more. Because now, by say adding protein, I have to reduce fat and or carbohydrates to keep the energy the same. Now, if we just change the type of fat, protein, or carbohydrate, that seems simple enough. For example, let's reduce saturated fat. Well, the more of one thing, for example, saturated fat, means less of something else. For example, polyunsaturated fat. So can we honestly say, with confidence, that whatever we observed was down to there being more saturated fat, or was down to there being less polyunsaturated fat? And remember, whole foods come with the vitamins and the minerals as well. So maybe changing from something that's saturated fat rich to something that's polyunsaturated fat rich changes the other nutrients. But even if we ignore that and we just focus on the macronutrients, as most studies do, there simply is no way of isolating only one thing. And as I said, well, what about the micronutrients? We're probably changing that too. Get most people into a study, irrespective of what it's looking at, even if they end up in the so-called control group where they get some kind of diet potentially not considered to be all that healthy, there's a good chance that by following instructions around eating, their health will improve. It makes the whole process of eating and food selection much more conscious and planned. Even monitoring, and some of you brought this up in your assessments, even having to record food intake made some people think twice, am I going to have this? Now, you may have thought of that because you feared judgment, or it may have just been inconvenient to add one more thing to the list. So you thought, I don't need it right now, I'm not going to have it right now. So that's another issue, and that is recording. So we're going to look at that next. So adherence and the details matter quite a bit. What was the method of assessment? Now, the food frequency questionnaire ranks rather low. This is basically asking you, how often do you have apples, let's say? And then it will give you some things to select from. More than once a day, once every 2 to 3 days, and maybe once a month, whatever it might be. That's a food frequency question, how often you have it. It generally won't ask you how much. So it's what we might call something that's semi-quantitative. It will get some idea of the quantity you're likely to consume, but it could easily be well off. And these have been heavily criticized because a lot of studies using them basically show that people's energy intake is inconsistent with staying alive. Those who defend food frequency questionnaires argue that, well, the goal is not to get an accurate amount, it's to put people in different categories. And they have a point. Next one is the 24-hour recall. This is exactly what it sounds like. Someone may ask you to recall what you had over the last day. And there might be various prompts involved to help you with the recall, or there might not. So you can do this at various qualities, but it's still a snapshot of only a single day. Then there's the estimated food record, could be called a log or a diary. This is what most people chose to do in their related assessment. So have an estimate. Now, of course, you could go one step further, and some people do and did, and that's the weighed food record. But again, weighing itself can interfere with how much people have, and sometimes it can be hugely impractical. If you cook a single meal for yourself and only yourself, and you have it all, that's the easiest option, but that's not the reality for a lot of people. Now, the single best way, and this is only possible for studies on quite a small scale, is the so-called duplicate diet. And you are literally asked to keep a complete duplicate of what you ate. So you would basically cook twice as much as you intended to eat, and you would put the other half in a bag for actual analysis in a lab later on. So this takes a lot of the guesswork and a lot of the errors in food composition databases out of the equation, but it's expensive, it's impractical, it's quite messy. By now, we have developed a few so-called biomarkers, biological markers. In other words, things we can measure in the blood, occasionally in the breath, sometimes in the urine, that research has led to us having a pretty good idea of in terms of what it corresponds to in the diet. So we can use some of these biomarkers to more or less verify what's going on. And a good example of that is if someone thinks they're on a ketogenic diet, then their blood ketones should be elevated, consistent with that. And we can measure that, and it's completely objective. Common limitations, well, basically listed them. They rely on recall, some of them. There is a tendency to over-report the good stuff, how many, well, or what people think is the good stuff, vegetables, fruit intake, things like that. Under-report calories, sweets, sugar, this kind of thing. Just outright misreporting, not necessarily intentionally. And then, well, we need to think about, was this technique validated in this group? In other words, was a better technique used to see whether the technique, the food frequency questionnaire, for example, you could validate that against a food record in a subset of research participants. So 10% might have to also fill out a food record, and then you'd make a comparison and you'd say, okay, so on the whole, the food frequency questionnaire, on average, overestimated this by 10% and that underestimated that by 5% and so on. So you want to at least look that some validation has happened. And on duplicate diet is very expensive to do. What about describing diets? This is another huge challenge. You would think it wasn't so, but it is. How low energy or low calorie is a very low calorie diet? Now this actually does have a reasonably agreed upon definition, and generally it's 600 to 800 kilocalories technically. How low energy is a low calorie diet? Now this, on the other hand, has no such consensus. So we can reasonably assume responsible people would use this whenever it was over 800 kilocalories. But that still doesn't help you. One person having 1,000 calories might not be that far off their requirements if they're a particularly small person and probably quite inactive as well. Whereas another person eating only 1,000 calories a day, that might represent a 2,000 calorie deficit per day for that individual. So that's another issue. How high protein is a high protein diet? Again, there is no universal consensus. So you see this term in a study title or in the newspaper, you really have no idea what it means unless you go back and you look at the actual numbers. And the same is really true for low carbohydrate diets. People who consider themselves intentionally following a low carbohydrate diet would probably define this very differently to those who consider low carbohydrate diets to be maybe less than ideal. It just depends very much on who is using that term and in what context. What about the Mediterranean diet? Researchers seem to have this generally broadly agreed upon view, but if you actually go to Mediterranean countries, you will find that their diets vary tremendously. Then there's another question that is even more specific to actual published research. Should macronutrients, so proteins, carbohydrates, and so on, be given in absolute gram amounts? So should we be saying that in a study, the average protein intake of participants per day was 72 grams? Does that make an awful lot of sense? Should it be given in grams per kilogram of total body weight? At least that way we can account for differences in people. And this is what we see in the nutrient reference values in terms of recommendations and outside the nutrient reference values as well. That maybe information is provided as grams per kilogram of total body weight. But then there are some who would rightly argue, well, okay, but is that the best way of doing it? It should be grams per kilogram of lean mass. Someone who has quite a high body fat percentage, even if their goal is to build muscle, won't need, let's say, 2 grams per kilogram of body weight, and they will probably be fine if they looked at something more like 2.2 or 2.5 grams per kilogram of lean body weight, or as it often is, should it be given in percentage of energy? Now, I don't have a particular view of which way it should be. Probably to allow for easier comparison between studies, ideally all of this would be provided, but it usually isn't. Just before I conclude, I'd like to say that despite plenty of people reciting the mantra that we should be eating a nutrient-dense diet, which basically means lots of vitamins and minerals per calorie or per 100 kilocalories, and despite dietitians and studies often working out ways for study participants to meet nutrient recommendations by making the diet more nutrient-dense, we don't generally see studies describing their diet as nutrient dense. There is an incredible focus on macronutrients and a lack of focus on micronutrients unless we're talking about supplementation studies. So let's briefly touch on that. Studying supplements. Well, the problem here is the question becomes, what was the form of the nutrient being supplemented? Not just what was the nutrient. Giving methyl tetrahydrofolate, sorry for the chemistry, but think back to when we talked about folate, is quite different to giving folic acid. Right? Folic acid isn't even active in the human body. It needs to be converted, but it is very bioavailable. So the form matters. There is some evidence indicating that the common supplemental form of vitamin B6, for example, might not just not be biologically active, but it might actually impair biologically active B6 activity. So it might be one study in cells, an anti-B6 that we find in supplements. How much were participants already getting in their diet? This plagues fish oil supplementation. If someone's already having a lot of those omega-3 fatty acids and then they add in a little bit of fish oil capsules, we wouldn't expect to see much of a difference. On the other hand, if that person was having hardly any of those omega-3 fatty acids and we add in a few fish oil capsules, I would anticipate seeing a much bigger effect. And yet often in supplement studies, the baseline diet is not really taken into consideration. Another thing is, what were body stores of that nutrient like? Were some people in the study deficient and others not? It's a bit like with that coffee example. One study that had a lot of people whose stores were low, or even on the deficient side, well, they're probably going to benefit tremendously. And if a study has a lot of those people, it's going to show how hugely beneficial the supplement is. On the other hand, if you have a study where most people already had basically enough, the supplement is not going to do them any good. Maybe it might even be slightly harmful. Then, how much of important cofactors were available in the diet or in stores? So remember, nutrients, especially micronutrients, well, don't work in isolation. In fact, we need the micronutrients to use the macronutrients. That's another thing, right? All of this is an intricate web, and unless we ensure we at least know what our participants were like, whether they had any major deficiencies, we really don't know what we're looking at. We know what question we're asking, but we don't know why we're getting the answer that we're getting. Physical activity studies, there's a lot of overlap with what I've just discussed. And of course, a key one is they often don't look at nutrient status. Right? If you add a lot of physical activity, and say in the form of exercise, on top of a diet that's far from meeting someone's requirements, it's just an added stressor. And I would anticipate people might actually make themselves worse. Not always, but sometimes. Was there dietary change during the intervention, during the study? So while people were doing whatever exercise or physical activity change that they were asked to do. It's not uncommon. Maybe the exercise made people feel better and that changed their outlook. And now they're choosing, not even necessarily consciously, to eat better. Or other people might go, well, I'm being more physically active now, and they might assume that that gives them license to eat more poorly, for example, or eat more, which generally it doesn't do. What about the actual physical activity before the study and during the study? Did that lead to a change in, say, non-exercise physical activity? Did it lead to a change in sedentary time? Did people, suddenly take up going to the gym, but spend the rest of their days less active as a result? Was that even measured? Baseline function, not just for outcome. How well did the person function physically to begin with? Often all we do, and I've been guilty of this myself, is maybe assess someone's risk in heart terms, in heart attack terms. So we might do a baseline test of fitness and we might monitor what's happening with the heart using an ECG, but do we actually also assess how well does this person move? How capable are they physically? And the answer is often we don't. Also, body composition, okay? Is that taken into consideration? And are we using good measures of cardiorespiratory fitness, or are we using relatively poor ones? Are we basing our assumptions on the International Physical Activity Questionnaire, or are we testing both various aspects of fitness, but then also using techniques like an accelerometer, for example, and maybe an activity log. Are we using any objective measures in this or not? Those are things to look for.